Welcome friends and welcome to my channel Sequential Biology. In this video we will discuss about reproduction strategy or the replication strategy of different types of viruses. Okay, and you know the viruses contains different type of genetic material. Actually different viruses contains different type of genetic material, and the DNA containing viruses, their DNA may be double stranded. Some viruses are double stranded DNA containing viruses, some viruses are single stranded DNA containing viruses. And show how these two types of viruses replicate we will discuss. Okay, and the RNA containing viruses are also two types: the single stranded RNA containing viruses and the double stranded RNA containing viruses, and which type of reproductive strategy they follow we will discuss it. Okay, and apart from this RNA, some viruses are plus ssRNA containing viruses, plus ssRNA containing viruses, and few viruses are minus ssRNA containing viruses. Then what is this plus ssRNA? When the RNA directly synthesize protein, then they are known as plus ssRNA, or they act like mRNA. Okay, and the minus ssRNA are those types of RNA which are complementary with this mRNA. Okay, so they do not directly synthesize any protein. First we will see how DNA virus replicate, and you know the DNA are double stranded DNA. Okay, so the DNA, the viral parental DNA, this is a viral parental DNA, and when this parental DNA enters into the host cell by breaking of capsid of this virus inside host cell by using the host cell enzyme, this DNA is released inside host cell. This is viral parental DNA, and first this viral parental DNA undergoes transcription, and the transcription is the process by which RNA are synthesized by using DNA as template. So this DNA actually undergoes the transcription, and this will produce an early mRNA. This will produce an early mRNA, and from this early mRNA the early protein are synthesized, and this early protein first block the host metabolism. That is, they hijack the total host protein synthesis machinery, and this early protein which synthesize first protein inside the host cell, they also serve as replicating enzyme, and with the help of this early viral protein the parental DNA will replicate, and this will increase in number inside host cell, and this progeny DNA contain late gene. Okay, so viral parental DNA contain early gene, and from this early gene the early mRNA will be synthesized by process of transcription, and from this early mRNA early protein will be synthesized, and this will block host cell metabolism. And also this early protein causes replication of this viral parental DNA, and this parental DNA form the many progeny DNA, and in this progeny DNA the late gene are present. From this late gene, by process of transcription the late mRNA are synthesized, and this late mRNA produce the late structural protein, and this structural protein actually form the capsid layer of this virus. So these are the capsid layer which are synthesized from this late structural protein. These late structural protein are from late mRNA, and which are synthesized from late gene. Okay, this late gene are present in progeny DNA, not in parental DNA, remember it. So this late structural protein and this replicated DNA or the progeny DNA assemble together and to form the nucleocapsid, or this will form progeny virus, and this is reproductive strategy of DNA containing viruses. The transcription of the viral parental DNA to form the early mRNA, and the translation form early protein. So this early protein is non-structural, that is they do not form any capsid. Okay, and this early protein actually shut down the host metabolism, and then this early protein helps DNA replication, that is the parental DNA replication of viruses, and this will copy, or this will produce or synthesize a huge number of progeny DNA, and this progeny DNA undergo transcription to form late mRNA, and this late mRNA translate to form the late structural protein. And this progeny DNA or this progeny DNA which are synthesized, or which are synthesized by process of DNA replication by using this early protein, and the protein material which are synthesized from this late mRNA are assembled together to form a complete virus particle. Then this is reproductive strategy of DNA containing viruses. Now plus ssRNA. Now we know that the plus ssRNA directly act as mRNA, and from this plus ssRNA the protein can be produced by process of translation. Protein can be synthesized by process of translation directly. So this plus ssRNA first they undergo the translation process, and this will produce the early viral protein, and this early viral protein acts as RNA polymerase. Early viral protein act as RNA polymerase. So from this plus ssRNA the minus ssRNA are synthesized. This plus ssRNA and this minus ssRNA are complementary. So this plus ssRNA are transcribed to form the minus ssRNA, and this minus ssRNA undergo the transcription process by using early viral protein. By process of this transcription, the plus ssRNA are synthesized, right? So this plus ssRNA are synthesized, and this plus ssRNA are not directly synthesized from this plus ssRNA, from the parental plus ssRNA. Actually, they are synthesized via early viral protein. This early viral protein acts as RNA polymerase, and this will causes transcription of this parental plus ssRNA, and from this parental plus ssRNA minus ssRNA are synthesized, and again the progeny plus ssRNA are synthesized with the help of this early viral protein. And from this progeny plus ssRNA the late viral protein are synthesized, and these late viral protein are component of capsid, okay, and then this progeny plus ssRNA and the late viral protein are assembled together to form the daughter variant. And this is reproductive strategy of plus ssRNA containing viruses, and poliovirus actually follow this strategy. So most RNA viruses, their ssRNA has some polarity as mRNA, and the RNA which have the same polarity with mRNA, they are called as plus ssRNA. They can directly translate into the early protein, okay, since they are plus ssRNA, so they can directly they undergoes directly translation process, and they will form early protein. The early protein acts as RNA polymerase. So RNA polymerase are those enzymes which are involved in process of transcription, and they direct the replication or the transcription of plus ssRNA. So plus ssRNA will be transcribed to form the minus ssRNA, and then this minus ssRNA will undergo the transcription process, and this will form the plus ssRNA, and from this plus ssRNA the late protein will be formed, okay, and this late protein are actually viral component, and this plus ssRNA and the late protein are actually packaged together to form the complete viral particle. Then this is a reproductive strategy of plus ssRNA containing virus. Next, minus ssRNA containing viruses. Remember, the minus ssRNA are not directly involved in process of translation. So it is very important for synthesis of plus ssRNA from this minus ssRNA. And minus ssRNA containing viruses are influenza virus, rabbit virus, and the myxovirus. Minus ssRNA is opposite polarity of the mRNA. So the direct translation is not possible, since they have the opposite polarity with the mRNA. Actually mRNA directly involved in process of translation, but this minus ssRNA has opposite polarity of the mRNA, so they are not directly involved in process of translation. In this minus ssRNA first transcribed into plus ssRNA. Actually they do not undergo the translation, but they first transcribed. So transcription is the process by which RNA are synthesized, but this transcription is performed with the help of viral RNA polymerase. Actually in viruses there may be present few enzymes, and this enzyme and the genetic material enters into the host cell. So this minus ssRNA first transcribed into plus ssRNA by using their own viral RNA polymerase, okay, and then this plus ssRNA must be undergo the translation process, because this plus ssRNA can directly involve in process of translation, since it has same polarity with the like mRNA. So translation serves as the mRNA, so viral protein are synthesized, and then this plus ssRNA also undergoes the transcription process by using the viral RNA polymerase, and then this will produce many ssRNA minus ssRNA. So many viral protein and this many progeny ssRNA will be packaged to form the many progeny viruses. This is a reproductive strategy of minus SS RNA containing viruses like influenza virus, rabies virus, and the myxoviruses. The minus SS RNA transcribed into plus SS RNA first using viral RNA polymerase, and this plus SS RNA actually involved in two processes. They translate into the protein. This is the structural protein, and they form the capsid. And the transcription of the template. So this plus SS RNA will be transcribed. So transcription is the process by which the complementary mRNA are synthesized from their template RNA. Okay. So transcription of the template undergo the replication to form the many copy of minus SS RNA. Then by this way, the minus SS RNA containing viruses, that is influenza virus and the rabies virus and the myxovirus, or replicate inside host cell. Next, double-stranded RNA containing viruses. The example of this double-stranded RNA containing viruses are reovirus and the rotavirus, and how they reproduce inside the host cell we will discuss. Okay. Now, first see the diagram. This is double-stranded RNA. So viral RNA polymerase causes transcription of this double-stranded RNA. Remember, this double-stranded RNA, since they are double-stranded RNA, so these two strands of this RNA are complementary with each other. So one strand of this double-stranded RNA form the minus SS RNA, and another strand will form plus SS RNA. Then this minus RNA and the plus RNA undergo transcription. Transcription is the process by which RNA are synthesized, and this minus RNA, by process of transcription, they will produce the plus RNA, because the transcription product is always complementary with their template. Okay. So since this minus RNA is template, then they will synthesize plus RNA. Okay. And many plus RNA are synthesized by this process, and this plus RNA are transcribed to form minus RNA, because this plus RNA are serve as template, and so from this template strand the many minus RNA are synthesized, and this plus RNA and the minus RNA are then form a double-stranded RNA. Okay. Since they are the two complementary strands, so they will be united to form the double-stranded RNA. Okay. And also this plus RNA, since they serve directly as mRNA, okay, so they will also produce the viral protein. These are the structural protein, and this structural protein and this double-stranded RNA are packaged together to form many progeny virus. This is reproductive strategy of double-stranded RNA containing virus like reovirus and rotavirus. So their double-stranded RNA are segmented, and each segment coding of one polypeptide. The viral RNA polymerase separate into the two strands. So viral RNA polymerase separate the two strands of this double-stranded RNA. One is the plus strand, which is sense strand, and this directly act as an mRNA. Okay. So the host ribosome synthesize the viral protein, right? So from this plus strand, the viral protein are synthesized by using the host ribosome, and also the minus strand and the plus strand act as template and synthesize the complementary strand to form the double-stranded RNA segment. Actually, the process by which the double-stranded RNA containing viruses reproduce or replicate. And in the next video, we will discuss about retrovirus reproduction strategy. So thank you, and that's it.